Today is the 31st session of the Bioanalytics Skill Development Forum's Journal Club and Networking Program, where our speaker for today is Dr. HARISHCHANDER ANANDARAM. So he is a assistant professor, senior grade from School of Artificial Intelligence, Coimbatore, and he will be taking a session on pharmacogenomics and miRNA regulatory networks in autoimmune disorders today. And So let me tell you a little bit about Dr. Harishchandra Anandram. So he completed his PhD in bioengineering in 2020, MTech in bioinformatics in 2011, and VTech in bioinformatics from Satyabhamana Institute of Science and Technology, Chennai. His research includes analyzing resistance in HIV protease inhibitors through molecular mechanics and machine learning, and exploring pharmacogenomics and miRNA regulated networks in psoriasis in collaboration with prestigious institutes like IIT Madras, Georgetown University, JIPMER, CIBA, and ILS. Dr. Anand Ram has published extensively in international journals on bioinformatics and systems biology. His work earned him Young Scientist Award from the Melinda Gates Foundation for his research on miRNA dynamics in infectious diseases. He has reviewed over 200 manuscripts in systems biology and is currently focused on predicting novel lead molecules and biomarkers to target inflammatory pathways in infectious and autoimmune disorders using computational techniques. So I welcome Dr. Anandaram to the session today.
Is visible to everyone. And let me put it in the PowerPoint mode. And it was the PhD topic, the pharmacogenomics and microRNA regulated networks in psoriasis. And it does involve mathematical modeling also. And for my reviewers, if you consider Dr. Jayakantan from Bioinformatics Department in Arakappa University and Dr. Karthik Raman from Biotechnology Department of IIT Madras. And why I've taken the topic is mainly because the prevalence of psoriasis worldwide gives you the gender percentage because it's twice more common in males due to the Y chromosome factor compared to the females, as per the Indian report. And most of the patients are in their third or fourth decade only gets diagnosed during psoriasis. And these studies are limited by the absence of commonly accepted and validated diagnostic criteria. Furthermore, there are no basic reliable information on time records of the disease, and the prevalence gets enhanced with respect to the 10,000 sample per one in the previous years. But the prevalence in India has gone from 0.44 to 2.8 as per the 2010 reports. Now it's even doubled as per the 2020 report. As you can see, the African Americans and Asians are in the range zero point 0.4 to zero point 0.7. And the prevalence studies from India are mostly hospital based, and psoriasis occur in 2% of the world population. And when you try to see the psoriatic march, how severe it is, and what is the cardiovascular drive which triggers psoriasis, you have the environmental trigger factor as well as the genetic factors. In some papers, even the factor like obesity and metabolic syndrome associated diabetes contribute to psoriasis to 5%, and the cardiovascular system dysfunction contribute to 8%. And then the psoriasis is also attached to psoriatic arthritis for the genetic and environmental predisposed analytics. And obesity is a risk factor for both psoriasis as well as the metabolic syndrome. And the inflammation associated insulin resistance based diplomia and the liver disease also gives the psoriatic march. And ultimately, the moderate purpose is to serve psoriasis directly and indirectly increase the risk of cardiovascular mortality. Psoriasis also precedes the development of psoriatic arthritis, and the selected key events in mapping the disease genes, because at present we work only on the post-genomic era, but the pre-genomic era also gave us the basic techniques like DNA blotting or FLP maps, linkage analysis, microsatellites, and extended linkage disequilibrium. And the key importance in those techniques are the DNA variant analysis, human genome initiation, and genome-wide linkage of analytics, and then mapping of many human disease genes, and the vision of linkage disequilibrium in mapping complex disorders. The detailed analysis of linkage disequilibrium in the human genome gives rise to the genome-wide association studies in hundreds of loci identified in complex disorders. And then when it comes to the recent development in the post-genomic era, from the single nucleotide polymorphism discovery to genotyping and the genomic sequence banking and the haplotype identification, from haplotype to the phenotype disease reaction to individual medical treatment. So the development of million SNPs coupled with the high throughput genotyping platform gives the comprehensive map of human haplotypes and the possible genome-wide association approach using cohorts or associated with the families. And when it comes to the NHGRI, the GWA catalog, you have the relative genome-wide association studies. You can see the published genome-wide study from 2011 to the initial quarter. You have only the basic HLA typing based chromosome markers, but you have the additional markers from the relative GWAS. As you can see, psoriasis is also in the catalog for the disease. And the abstract of the current work involves the microRNA to get enhanced. And I have used the computational systems biology technique with four computational approaches, mainly top-down, bottom-up, direct, and indirect. And a core objective of these approaches is to identify a potential biomarker to diagnose and treat psoriasis in future. Among the overall identity and the comparative analysis, we have identified that the HSA, that's the human microRNA 125A3 prime marker, was highly compatible with regulatory mechanisms with proper binding free energy computationally. And the objective is to get the list of top-down approach from PubMed, OMIM, DisGNet, and String, and analyze psoriasis with respect to the RNA-seq data based on Array Express as well as the NGS data and the string data and pharmacogenomic based data from Pharmaco, MAR, DBSNP, Form GKB, and string. And then the translational regulation based on the studies from TransFac, RAIN, and the interaction analysis of protein and string. And we do identify the predicted and validated microRNAs for psoriasis associated genes from target scan, Meritar based pharmaco-MAR, along with the interaction and analysis of proteins in string. And we also construct the regulatory network of genes associated with psoriasis by cytoscape. And then we also identify the key microRNAs in the regulatory networks by plugin of cytoscape, MIRMAP, and miRNA triplex. And we do analyze the pathways associated with pharmacogenomics of psoriasis associated with top-down, bottom-up, direct and indirect approaches. And the literature review key points reveal gene regulation initiation in 2007 and significant progress of microRNA morphogenesis in 2007 and the skin lesions in 2010, the recent studies on expression levels in 2008, structure of basic gene regulatory network in 2015, and the extended framework model in 2014. So the method I have used, as I told you, in top down, it's only the database of DisGNet or MIM, and then the mRNA from Meritar base and the transcription factor from Reg network and the construction of regulatory network in Cytoscape and identification of the top 10 regulatory network nodes in Cytohubba, and then the gene regulatory network relative analytics, mapping further. Secondary analysis is being performed in the microRNA triplex, which contains two microRNAs and gene regulatory pathway. And we retrieve and analyze the associated pathways in David. And we also identify the role of microRNA in translational repression. And in bottom-up approach, we used the differential expression data set from RNA-seq and GeoB or Array Express, the NGS, as well as the microarray data, and rest is the same for the process. And in direct approach, we do worked on the pharmacogenomic data from the PharmGKB database, and we also correlated with the pharmaco-MAR. And further secondary analysis with the same map and MAR regulator networks. Finally, the David annotation tool for the pathway analytics. And then when it comes to the method, it's the indirect approach with the identification of transcription factors associated with psoriasis from Mogrify and analyze the genes and microRNAs associated in psoriasis from database. And we do construct and analyze the network in Cytoscape. And from the Cytoscape network, we get the top 10 regulatory nodes from CytoHub. And then we identify the implication of microRNA and regulatory networks in MIRMAP and mRNA triplex to get the associated genes and pathways in David. And And when it comes to the result part, as you can see, though there is initial seed pairing of microRNA, there is no complete binding. Only in certain cases you have the complete binding, and only in one or two cases you get the nature of the triplex formation of the microRNA and the gene. And then whatever we have done is computational and rational based statistics. But whether do the computational and rational based statistics do have a meaning in the experimental results we do or also being carried out? Okay, when it comes to the results part, when you get the scope and significance of microRNAs as well as the transcriptional regulatory network of psoriasis, in the top-down approach, you have the combinatorial analysis of microRNA-based regulation in the relative points as well as the transcription factor, the relative activation binding. Then when it comes to the discussion, because all the results we got from our initial results were were already reported, and then we have fine-tuned our research into the next level. So STAT2, IL-17, CCL5, CCLX11, and relative sites were already reported. And the scope and significance of microRNA and translational regulatory network were also analyzed, not only the transcriptional network. And then we do discuss certain key regulatory factors and proteins associated. One novel result we got in translational regulation is the ACTL8 marker, which is yet to be explored in experiments. So it can be a good part for the experimental biologists to take it. And then the relative protein-protein interactions do give the biological pathway. As you can see, there are a lot of connections which are not been inbuilt. How to build the connection and reconstruct the pathway is also an additional factor. And then the annotation of KEC pathway with psoriasis gives the relative module. And then the mathematical modeling of microRNA-based transcriptional repression and regulatory network of psoriasis in top-down approach gives the mathematical modeling to understand the network dynamics. And the gene, microRNA, and transcription factors gives the rate analytics. And regulatory networks do follow the differential equation. So the rate of synthesis is associated with the rate of degradation, and the second order differential equation to get the above mentioned STAT2 and SP1 format in combination. And then the first stage node of transcriptional regulatory network of psoriasis gives the bottom-up approach of miRNA-based regulation, and the transcriptional regulatory network with the gene Wi-Fi involves markers, DMNC, MNC, and clustering coefficient methods. And there are other transcriptional regulatory network of pure DM1, STAT3, MEC, MXD1, MMP9, NM1, HSP4, and SYNCRIP. And the implication of microRNA in transcriptional regulatory network of psoriasis gives the microRNA-based co-expressed regulation. And the significance of expression-based transcriptional regulatory network of psoriasis gives the microRNA and transcription factor binding in the gene. And the CTCF in gene is the additional factor getting associated with the network. Then the translational regulatory cascade is associated with the microRNA and the protein module extension. And then the protein-protein interaction network of psoriasis in bottom-up approach gives the expression-based regulation for the primary as well as the secondary analysis. And then the co-expression based regulation in bottom-up approach gives the node degree values as well as the extended pathways. And finally, the mathematical modeling of microRNA-based transcriptional repression in regulatory network gives the additional factors. So the gene, microRNA, and transcription factors gives the relative modeling of the reconstructed pathway based on the differential equation. And when it comes to the direct approach, already reported SNPs, loci, alleles, and genes were taken. And you can see the genes involved in pathogenesis and their additional regulatory network. And then you do have the genes and the possible comorbidities in psoriasis and the co-expressed modules of psoriasis and rheumatoid arthritis, and the gene subsets shared between the comorbidities of psoriasis and arthritis. The gene subset shared between the comorbidities of psoriasis and related arthritis, and the subset of genes involved in cellular communication, the genetic variations, critical proteins involved in psoriatic cell communication, and in the direct approach, the transcriptional regulatory network gives the microRNA and the gene association, and the associated regulatory network form this the clustering coefficient. And the significance involves MTHFR gene, Shasame 125 3P microRNA and AHR gene. And the relative activation of MTHFR by AHR and the additional MTHFR gene for two binding sites with AR and T gives the additional module. And in the direct approach, we get the translational regulatory network of the specific microRNA, which was not reported initially, and we are the first to report it. And in the indirect approach, we get the combinatorial modules of the microRNA gene and the transcription factors, and the top 10 hubs in CytoHub in the indirect approach. And then we do have the implication of canonical triplex for only the specific microRNA duplex and the gene singlet formation. And next, the annotation of KEC pathways based on the p-value and Benjamini statistics. So HTLV1 infection is also associated with psoriasis. And then till now we have seen in the molecular level and the system level how it really works is we take a condition of pathogenesis called epidermal hyperplasia. So the role of interleukin 23 in psoriasis pathogenesis is trying to be modeled mathematically. So the triggering of dendritic cells is happening by the tumor necrosis factor, and the interleukin 23 stimulates the resin 17 to produce the tumor necrosis factor alpha and the interleukin 17. The tumor necrosis factor alpha and interleukin 17 work synergistically to stimulate the keratinocytes production by cytokines, chemokines, and the antimicrobial peptides. The chemokine recruits neutrophils and CCL20 to recruit the T cells and the dendritic cells. And the mathematical modeling involves the identification of the role of cytokines in the key components of the pathway. And we do use variables to mention the key components and formulation of equations for the desired entities. And we evaluate by resolving the system of equations with the desired value. As you can see, since these are the major factors, they are named as X, Y, and Z, and the initial concentration is named as A, and the rate of stimulation of tumor necrosis factor alpha and interleukin 17 producing antimicrobial peptides is alpha 1 and alpha 2, and the rate of stimulation of TNF alpha interleukin 17 to produce chemokines is beta 1 and beta 2, and the rate of stimulation of TNF alpha and interleukin 17 to produce CCL20 is gamma 1 and gamma 2. And K1 is the rate of influx of neutrophils to side the inflammation, and K2 gives the rate of influx of T cells to side the inflammation, and K3 gives the rate of influx of dendritic cells to side the inflammation. And mathematical model results in forming the reconstructed pathway base and using ANFIS, the mathematical model of IL interleukin 23 mediated pathway is being taken. So it is identified that a major role is played by neutrophils, T cells, and dendritic cells. And furthermore, the model is subjected to experimental conditions, and we have computational research along with mathematical modeling. So the reconstructed gene, that is transcription factor, and the microRNA gives the transcriptional regulatory network of psoriasis, and the pathway is reconstructed now. And the subnetwork analysis is also being obtained by using the molecular complex detection technique, clustering technique with a cutoff of 2 node and the relative scores and statistic. And then whether the transcriptional regulatory network is really playing a role in the pathway, and any experimental proof is there for our predictions and all. Already there are experimental proofs for our prediction in pathway level. So this database gives the experimental orientation of the predicted pathways, and you can see the disease association ratio is there already for the pathway based on the molecular cascade of mechanisms. And finally, we consolidate the genes mRNA transcription factor network, and we do identify the hubs in the network and the relative microRNA and the relative associated factors. And then when it comes to the biological process, it's the signaling pathway which get extends for the EB or model. And then when it comes to the relative pathway, it's tyrosine kinase. And the study has just aided an initiation to understand the deciphering complex of regulatory mechanisms involved in pathogenesis of psoriasis, and will provide putative and therapeutic targets more. And further investigation and validation of the obtained microRNAs and transcription factors associated with various biological pathways involved in pathogenesis of psoriasis require more experiments. So the scope of future work is simulation of regulatory networks from mathematical model to identify the significance of microRNA in disease pathology. So this is a recent publication from our group, and these are the initial publications. These are some of the papers presented in actually, these are the results we have from various papers. You can see the pharmacogenomic based regulatory network and microRNA association, and then top-down approach, and with the Meritar base, and with respect to the mirror map and with respect to the top-down approach and mirror base and bottom-up approach, the relative bottom-up approach, and then the microRNA transcription factor in the association. And these are the major queries raised by the foreign reviewer for a paper. If I would like to draw a conceptual or a theoretical framework, what would it be like? A pipeline development for regulatory analysis, entirely based on silicon computational and expert. What's the key finding? The key finding is performing a pilot study once again with the initial phase with the minimum of 10 clinical samples. What kind of biomarker with significance for the HSA MIR 125-3PB diagnostic? Obviously, it has to be a therapeutic marker. Already, it is a therapeutic marker associated with the polymorphism in cancer, but not in psoriasis. So, for psoriasis, it can be tried. And then you do have papers signifying the fact that the phosphoinositol 3 kinase and AKT signaling pathway association and the relative target extension. And from this study, is it possible to identify the exact link between psoriasis and arthritic and streptococcal infection or psychology? Yes, we do have the gene regulation study along with environmental factors to do the modeling for identifying the correlation between the infectious and autoimmune diseases on the basis of network motifs in the hub and comorbidities are also being analyzed. And what's the purpose of ordinary differential equation for mathematical modeling is the query raised by the Indian examiner. So to explain the activation of transcription factors and the repression of microRNA in gene regulation, and to explain the activation of interleukin 23 in inducing epidermal hyperplasia in psoriasis through the activation of cytokine chemokine receptors in the pathway of NF-kappa B. Are there certain network parameters more promising than others in identifying important targets? Yes, the degree parameter is more promising in identifying the key regulators involved in the pathway associated with the disease pathology. And how do you extend this to other diseases? Can it become a pipeline? This work can easily be extended to other diseases because all diseases occur due to errors in autoimmune mechanisms on the basis of improper signaling in gene regulation. This study can become a pipeline for the analysis of microRNA based regulation. And finally, the Indian examiner asked me what would be the most promising way to tackle psoriasis based on the thesis. The most promising is the biomarker HSA MIR 125VA3B because already it's a proven marker for cancer and the same can be applied to psoriasis with some significance. And you do have polymorphism based study for the marker with the MTHFR gene and the mTOR signaling pathway with phosphoinositol 3 kinase and AKT signaling. So further discussion can be done based on the process. Okay, you can ask your doubts, students. I'll help you. Yeah, when it comes to the top-down approach, it's directly the data present in the database. And when it comes to the bottom-up approach, it is the differential expression-based data where additional analysis need to be done. When it comes to the direct and indirect approaches, it's the pharmacogenomic data and the correlated data already present in the database. I hope I've answered your question. Welcome.
Excuse me, sir. So, which
Yeah,
approach
please.
is suitable? It's used frequently, top down or direct or indirect approach.
Well, it depends on the data because when you try to do a pathway reconstruction, all data are required. Because when it comes to differential expression study, it's a particular time. That data alone won't be enough. When it comes to pharmacogenomic data, it is for one association. That also won't be enough. So when it comes to identification of a biomarker, all data and all approach to be done. If you want to see only one particular marker at one particular time, differential expression analysis alone is enough. Have I answered your question?
Yes, sir.
Students, you can ask your doubts to sir here. He'll be happy to take your doubts, and also you are
Mm.
you have access to mic, and you can even turn on your videos if you want. Even write in the chat box. So we'll wait for 5 minutes. If there are any doubts, you can list them out now.
Well, when it comes to the correlation development and measurements, yeah, it is done. Initially, it was the clinical examination of lesion by PASI score, but now they are trying to use biomarkers. But it's only in the phase two clinical trials. In cancer, it's already in phase three and going to be approved soon. Well, when it comes to curing, furthermore experimental studies need to be done. As I've told you, it's only in phase two, not in phase three. Have I answered your questions? That's Sushen Mandal and Saina. I hope your questions are being answered. If you have further doubts, you can ask.
So I believe there are
Yeah,
no more
please.
doubts.
Okay, sir. Fine. Yeah.
Yeah.
And
So,
thanks a lot for the opportunity, sir, to share my work and interact with students. And thanks for the patience also. Initially, the screen could not be shared, and then it was rectified.
in a
Thanks for
virtual
the
session,
support.
these are some issues that we have to, you know,
Yeah, yeah.
take care. Like there are anyways sometimes like technical issues. Like even our
Yeah,
host was
definitely. It
unable.
is from my side also because some internet connection was low from my side initially. Then it was rectified. And thanks a lot for the patience. And your team helped me a lot.
No issues, no issues. So today's session on pharmacogenomics and miRNA regulatory networks in autoimmune disorders was really insightful session, and I hope that participants will also agree with me. And thanks to you, Dr. Anandaram, for this wonderful session, and also to the
Thanks a lot, sir.
participants for joining and interacting with our speakers. And I hope it has been a wonderful and mutually benefiting experience for both the parties. And I also want to thank the like supporters of this student general club program for the constantly supporting this initiative, such as ACS IIT Bombay, IIT Delhi, and BioX Club and other student bodies. So I sincerely thank them for their support. And yeah, thanks to everyone. And I hope you have had a great day, uh, great learning experience. So
Thank you,
Saina,
sir. And there is one hand raised, if you could notice.
so Saina,
Yes, sir. Good evening.
Hmm.
Sir, I had
Yeah,
a
please, ma'am.
question that,
Please, ma'am. Please.
sir, for all this work seems very interesting to me. So, I had a question like, can we have a visit to this place?
Which place?
Like, where you people work to have like offline, we see all these things.
Actually, it's a multi place work because for differential expression, experimental works, I need to travel for four places. One is JIPMER Pondicherry, one is IIT Madras, and one is Institute of Life Science Bhubaneswar. It's not a single place work because in Institute of Bhubaneswar only you have the animal house to get the in vivo data, and IIT Madras you do have the molecular based data, and in JIPMER you get the clinical data. But you are always welcome. If you are
Oh,
interested, I will
okay.
put a word to the people who are working there, and whenever they are free, you can visit those places.
Sir, also one more question. So currently I'm a pharmacy student. I'm in second year.
Hmm.
So is there like we can also work under this pharmacogenomics and this miRNA for the research
Definitely,
related
you
work?
definitely you can work.
Okay,
Definitely you
fine,
can
sir.
work.
Like
If it
as
is
a
only an in
clinical
silico work,
pharmacist?
yeah. If it is only an in silico work based on the already present data, you can do it in online itself. If you want to do experimental work also, then you need to visit those places, get experimental stuff, and do it. I hope in your college also basic experiments can be done, right? Cell lines, and because being a pharmacy department,
Yes, sir.
Saina, yeah, you will be having cell lines, no? So you can test it on cell lines, right? Based on the simulation results.
Yes, sir.
Yeah, it can be done.